So we've got about an hour to finish up the lecture on acquired immunity. And where we left off is really exploring these T-cell receptors, right? So we described B-cell receptors, which are these immature IgG molecules that are tethered to the membrane that will eventually become a full secreted IgG. In the case of T-cell receptors, there are these TCRs that are going to bind to and recognize peptides that are displayed on professional antigen-presenting cells, and non-professional antigen-presenting cells as well, through this major histocompatibility complex, either MHC class 1 or MHC class 2. And where we left off was this crystal structure where you see, in this case, a TCR. Here is the MHC class 2, and here's the peptide motif that it's displaying to the T cell. And because this is MHC2, it's going to couple with CD4 positive T cells, right? So the cute little math analogy that we made, or math equation for helping to remember which MHC pairs with which CD4 molecule. And we followed, or we ended here with CD4 and or CD8, that it's not interacting specifically with the peptide or the TCR, but actually the MHC, either MHC class 1 or MHC class 2. Okay. So as a reminder, MHC class 2s are going to be expressed and presented on what we call professional antigen-presenting cells, right? So these are going to be your B cells, those dendritic cells, and your macrophages. And MHC class 1 are going to be presented and expressed on every one of your cells, okay? And that'll become important as we talk about how immunity arises. All right, so we've talked about then, in this case, TCR receptors are going to be presented on MHC2s or MHC1s. And a question actually arose during office hours, and here I'm going to go into it, is how does the peptide actually wind up getting out there on the MHC? And there are really two core mechanisms. One is the exogenous pathway, and we're going to talk about that here right now. And the second is the endogenous. Okay. So in the exogenous pathway, what you're going to have is, this is a professional antigen-presenting cell. So again, a B cell, macrophage, dendritic cell, right? And these are professional phagosomes as well. So they're going to recognize something about that foreign element, usually a bacteria, but it could be a virus, or a foreign cell of some type, and it's going to engulf it, right? It's going to phagocytose that particle, let's say it's a bacteria, and it's going to isolate it in what's known as a phagosome. This is just an endosome, right, that has the entire bacteria inside of it. When the phagosome is going to fuse with a lysosome, right, we talked about all this before, in order to subject that bacterium to a host of enzymes that are going to degrade that bacteria into its constituent pieces. Okay, so it's going to digest that phagocytosed antigen using lysosomes, right? And what it's going to do is then these digested pieces here, this vesicle is going to fuse or combine with another vesicle that actually contains those MHC2 proteins, MHC class 2s, okay? So I've got a vesicle that's got MHC2, I've got this vesicle that has all these digested bits of a bacterium. Those are going to fuse, and now what I can do is the MHC class 2 has access to all those peptide motifs in order to bind them, okay? And this is where peptide loading is going to occur. So it's going to occur in these little endocytic vesicles. That vesicle is then going to be transported to the plasma membrane. It'll fuse with the plasma membrane, and as a consequence now have MHC class 2 with a peptide that's loaded on the outer cell surface, right? It's going to interact then with, in this case, a CD4 positive helper T cell, right? So helper T cells are the ones that are expressing the CD4. CD4 is the protein, remember, that's going to help couple with MHC class 2s, all right? And so in this paradigm where we have MHC class 2 with an epitope bound to its cognate receptor, right? So this, not only does a TCR receptor, the TC receptor, have to engage with MHC2, it has to recognize that peptide motif. If that occurs, you get CD4 binding, you promote the activation of this helper T cell, and a number of downstream events will then occur. One of those is the expression of cytokines, right? Okay, so it's going to express cytokines. It's going to, in the case of helper T cells, this will be primarily interleukins and interferons. These are just subclasses of this broad class of proteins known as cytokines. And these can interact with certainly macrophages in a paracrine fashion, or they can act in an autocrine fashion, right? So these cytokines can actually impact and promote the further maturation of that T cell, that helper T cell, to amplify signal, right? To perhaps amplify its expression of cytokines. It can actually, those cytokines can act back on this antigen presenting cell, and depending on what that antigen presenting cell is, can have various impacts on that. I'll highlight that if this is a B cell that has engulfed, that has recognized through its B cell receptor, has recognized an antigen, caused the phagocytosis of that particle, right, that bacterium, and now is presenting it to a helper T cell that also recognizes a peptide within that gamish, what'll happen is those cytokines will actually potently stimulate the proliferation of that B cell, right? It's a very strong, potent amplification signal, right? Because what it means is it's like a double, we were talking about logic gates earlier, a double logic gate, right? The B cell has recognized the pathogen, and a separate T cell has also recognized that pathogen. Chances are that is a really important thing, right? That B cell has recognized something that's very important, right? And so that autocrine loop, or that paracrine loop in that case, will potently stimulate the proliferation, the doubling of that B cell, okay? All right, so it's one way to think about the exogenous pathway is exo, right, is outside, right? And so these peptide motifs and the bacteria, right, these antigens, never actually access inside the cell, right? And so if you remember when we talked about membranes, this idea that the inside, the lumen of a vesicle that's inside the cell is still outside the cell, right? Meaning it does not have access to the cytoplasm of the cell, right? So the cytoplasm is the inside of the cell, and in this case, the bacteria is pulled in. It's still isolated in an exterior environment. It's degraded, it fuses, and then it gets pushed back out to the outside of the cell, right? So never having access to the interior or the cytoplasm of the cell. And that's going to be in contrast to the endogenous pathway, right? So there are lots of things that we've even talked about. Bacteria, right? Listeria has a nice little way to get itself out of early endosomes or phagosomes and burst into and gain access to the cytoplasm. A number of viruses are also going to evade this sort of normal phagocytic process. In many cases, they'll actually induce their own internalization and then escape these endosomes, right? And so we have to have a mechanism in that case in order to deal with some type of pathogenic or antigen species that are in the cytoplasm. And how we deal with that is through MHC class 1. Right? Now this MHC class 1 are expressed on all cells, right? So a virus in this case is not being phagocytosed by necessarily a phagosome, but rather infecting a normal cell. And this is also the process that's going to engage our own immunity against cancerous cells, right? Cells that have now all of a sudden expressing some type of mutant protein that they shouldn't be making, right? During normal protein degradative processes, these peptides will also be displayed out on the surface of these cancer cells. And it's in this way that CD8 positive T cells or the cytotoxic T cells will discover them. All right, so virus infected cells and or cancerous cells are distinguished by the proteins they make themselves. And these viral or self-proteins or peptides, right, peptide motifs, are in this case going to be pumped into the ER by an ABC transporter, right? So if they're in the cytoplasm, somehow we have to get them across a membrane barrier so that when they're so that they can actually be presented on the outer surface of the cell. And so in this case, it's an ABC transporter. We've talked about this broad class of ABC transporters in the past, right, that can transport large proteins and or peptides across the membrane. And in this case, it's going to pump these peptides into the ER. Now I'm inside the lumen of a vesicle. It confused with these other exosomal vesicles, right, that contain MHC class 1, that can be peptide loaded. And now when they go to the surface, they're displaying them out on their cell surface. Your cells are doing this all the time. So you're presenting self-antigens and pathogenic antigens all the time to your immune cells, okay? Even the ones that are normal, right? That's all these things do, is just present protein peptides to T cells. And all right, so here it is in pictorial form, right? So we get a CD8 positive or a cytotoxic T cell that recognizes MHC class 1 with its epitope, co-binds with CD8, right? And as a consequence of activation of a cytotoxic T cell, the cytotoxic T cell will start expressing cytotoxins, right? These are the porphyrins, right? Those proteins that start to form pores in cells to allow their contents to be released. And can also be enzymes, peroxisomes, like things that will help destroy the cells that are in the neighboring zone. Okay. So are there any questions about this to this point? Yeah. Yes. Yep. So cytotoxic T cells are defined by their expression of CD8, right? This is the way to do the logic. CD8 will only bind MHC1, right? Correct. Yeah. Helper T cells express CD4. Yes. So yes, they're always expressing CD8. There may be, right, this is a pretty simplified, and you'll find out here at the very end, there's even a subset of helper T cells that I'll talk about that are restricted to certain kinds of these CD proteins, right? And so CD8 defines the cytotoxic T cell. They may express other adapter proteins that modulate and specify their response a bit more, okay? All right, so where are we now? So important facts. One is, each antigen receptor, regardless of whether it's a B cell or a T cell, is going to recognize a very small number of structurally related epitopes. This is this concept of one antibody, right? One B cell receptor will bind one epitope. One T cell receptor will bind one epitope. I'm going to flex that a little bit and tell you that there's some flexibility in that, right? They're either the same peptide, only one peptide, but small modifications of that peptide might also be recognized, right? So it's a very narrow epitope space. And so what I would say is that the epitopes have to be extremely structurally similar, okay? And so this is based on a lock and key mechanism that we've talked about in the past, about how proteins sort of lock into place with their cognate receptors. Regardless of the type, again, B cell or T cell, once that cell is differentiated, each cell is committed to only producing one specific antigen receptor. One B cell will produce one antigen receptor that will eventually be turned into one soluble IgG molecule, right? Many of that, but only one. And that is forever, right? That gets hardwired in the system. So if you combine these two, this explains how we get this high level of specificity from both B cells and T cells. Yeah?
So I understand that one cell can only produce one specific antigen. Is it
receptor.
Antigen receptor?
Correct.
But can multiple cells create that same receptor?
Yeah, so what you've asked is, can multiple cells express that same receptor? The answer is yes, because it is possible to get clonal expansion of these cells, where it's the same parent cell, but it doubles into multiple daughter cells, right? The reality is also that you can have two, let's say, just say a T cell, you can have two T cells that express a T cell receptor that also can perhaps both recognize the same epitope, but maybe through slightly different mechanisms or affinities, right? Like how it's interacting, that antigen epitope is interacting with T cell receptor, may be regulated by different, kind of slightly different chemistry, okay? And we're going to talk about all of this diversity here in a second. All right. So we get highly specific immune system to, again, one lymphocyte is going to target each epitope. Okay, so if this is the case, and we have to be able to defend ourselves against both known and unknown pathogens, perhaps even pathogens that don't exist today, right? How does that happen, right? And so you can imagine now the number of lymphocytes, B cells and T cells, you have to have in your body right now that are ready to respond to unknown threats, right? So there's a huge variety of antigen receptors needed to provide this level of diversity of our immune system. All right, so we're going to talk real quick now about the cellular origins of diversity. In the very next lecture, which is on Thursday, we're going to start talking about the central dogma, how genes arise, right? How they're then turned into RNA and protein. And that is going to constitute, and we're going to couch that in the light of the molecular basis for the amount of diversity you have in your immune system. But what I want to talk about today is at a slightly higher level, is once we've generated that molecular diversity within this lymphocyte population, how does that cell population maintain its diversity in a way that's healthy and safe for us, okay? All right, so we're going to talk right now about the genesis and diversity of lymphocytes, acknowledging that the molecular is going to come on Thursday. All right, so when we think about the differentiation, let's say, of a B cell into a plasma cell, right, the professional antibody-producing cell, right, that effector cell of the B cell, right? When we think about that differentiation, it's really going to occur in two phases, right? One is an antigen-independent differentiation phase, right, where I'm not worried about exposure to any antigen, right? And the second is the antigen-specific or antigen-dependent differentiation. All right, so let's talk a little bit about the origins and the precursors for these cells. So when we think about the precursors for all lymphocytes, if you remember that cellular differentiation tree, it all started with the hematopoietic stem cell. That's this HSC right here. And it's within the bone marrow, it's going to differentiate into what we call the common lymphoid progenitor. Again, this was in table form in a couple of slides that we've shown. What that common lymphoid progenitor can do, as you can imagine, is differentiate into both a B cell and a T cell, okay? And so the B cell is actually going to be differentiated still within that bone marrow. The difference is that a T cell is actually going to be differentiated in the thymus, right? So another easy way to remember where B cells and T cells are differentiated, B cells are differentiated in the bone, T cells are differentiated in the thymus, right? So the first letter kind of helps you to figure that out. All right, anyone know where the thymus is? Here? That's the thyroid. Oh, that's the hypothalamus. Lots of THYs. I love it. Guess what protein I did my PhD on? Thy1. God, the thigh just keeps on showing up everywhere. All right. It's actually right here in your sternum. Well, or below, behind, right? So it's a glandular tissue. It's behind your sternum and between your two lungs. Okay? All right. So once, what is in this hatched box here is all of our antigen-independent differentiation. What this antigen-independent differentiation is going to generate are what we call mature, naive lymphocytes. I know that sounds a little bit wonky, right? Mature in the sense that they're committed to producing that one receptor, right? But naive in the sense that they have not interacted with their antigen, their cognate antigen, okay? Where all of our antigen-dependent differentiation is going to occur are in these second lymphoid tissues, or secondary lymphoid tissues. Okay? This would be your tonsils, right, that most of us have removed, your peripheral lymph nodes that are all over, get bit by a tick. I found this out when I owned a farm and my kids would get bit by a tick. Their lymph nodes back here get swollen because all the goo comes down. Yeah. So the lymph tissue is where you're getting all of this filtration of bodily fluids, and they're running over these tissues that are chock-a-box full of B cells and T cells, right? Because they're sitting there waiting on fluids and peptides and antigen-presenting cells to flow through, to migrate through these and try to present antigens and detect antigens that they need to attack. It's a really wonky thing. I was like, wow, why does my daughter have a bump on her back of her neck? And the doctor said, did you check her head? And it was like a big old fat tick. And I was like, oh, I feel like such a bad parent. All right. How did I not detect that big old fat tick on the top of her head? We lived on a farm. Yeah, she's a rough and tumble girl. All right. Okay, so let's talk really quickly then about this antigen-independent differentiation. All right? So it's going to follow this basic premise that stem cells, those hematopoietic stem cells, are going to give rise to many millions of different antigenically committed lymphocytes, right? These are the receptors, the B cell receptors or T cell receptors. Each one produces one specific antigen receptor, I've said that, generated at random. And again, we're going to talk about how that occurs in the next lecture. The generation of naive lymphocytes happens before birth in the primary lymphoid tissues, the bone marrow, or again, the thymus, and does not require the presence of an antigen. Okay, so we're talking about generating a number of these lymphocytes that are all going to express their own cognate receptor. Okay, and so you can think about this as a way that the body generates this absolutely enormous library, right? A library of potential receptors that are capable of, if you can find them, right, of possible matches. As you can imagine, much of that is absolutely useless. You'll never actually utilize those cells, right? Because you may never interact with a pathogen that has a peptide epitope that T cell or B cell receptor binds, right? Okay. You can imagine also, though, if I have this huge diversity and they're randomly assigned, and we'll talk about how they come about, but on those, let's say the antibodies, the B cell receptor, those loop structures that are on the very tips of those Y structures, that's where you randomize it. Right? And I have a real problem if I have a B cell receptor that can bind to self-antigen, right? So much of it is absolutely useless, and some of it's extremely detrimental to our own liveliness, our livelihood. All right. Okay, so we're going to take T cells as an example, but B cell pruning, this process known as immune pruning is going to be extremely similar. In the case of T cells, they're both positively and negatively selected in the thymus. And again, this is before birth, right? So in utero, you're going through this process. The first selection is a positive selection step. And so what we want is to make sure that those mutations that we are introducing on the T cell receptor itself that we haven't somehow destroyed the ability of the T cell receptor to actually bind to MHC, right? If I have a T cell that cannot bind to the MHC, right, either one or two, depending on the cell type, then there's no way that T cell will ever generate a cell immune response. Okay. All right. So we're going to keep only the T cells with TCRs that bind adequately. In this case, this blue and this purple, to MHCs in self cells. So here's a thymus cell, and there's many, many of these cells in the thymus. Again, it's chock-a-box full, and the T cell, all these immature and naive T cells are bumping in and interacting with each of these cells as they sort of percolate. You can think about it maybe like percolation. It's a little more active than that. But as they migrate and move through this organ system, they're being interacting with numerous thymus sites. Okay, and so the rest of these, let's say this red T cell, for whatever reason, the mutations that we've generated naturally have caused that T cell receptor to no longer bind MHC or any self-peptides that you're on there. So what we're going to want to do is kill all those. So what this thymus cell is going to do in this case is provide a survival signal. Anything that doesn't find a binding partner, right? It's like moving day, find your moving partner. If you don't find your moving partner, you get left behind, right? In this case, you don't get a survival signal and you die. So that's positive selection. Then they're going to go through this process of negative selection. Well, I want it to bind, but if it binds too well, then it might be a little too promiscuous. And I don't want this thing binding to self-antigen, right? So now it's going to go through this process where any thymus cells, right, these are, again, professional antigen-presenting cells that are presenting all of your self-peptides, right? Every single peptide motif that you can generate from your own body. If the T cell receptor binds to that too tightly and becomes activated, what is that? What is that the very definition of? Autoimmune, yeah. That is the origins of autoimmune disease, right? Is you've got an immune cell that recognizes self and thinks it's foreign, okay? So in that case, T cells that bind too well to self cells, we're going to generate an apoptotic signal and try to kill those, all right? So in this case, we have bound to blue, and so we're going to kill that one, all right? What survives this process is less than 5% of all those cells that you generated, right, in your bone marrow before you were ever even born. 95% of those are gone. Actually, greater than 95% are gone, right? So we've sacrificed diversity, right, but for a good cause, right? We don't want to generate self-responses, okay? And B cells are going to undergo a similar, again, it's often termed immune pruning, okay? We're trying to get rid of any reactivity to self-antigens. All right. So this is like the Goldilocks, right? Can't be too hot, can't be too cold, has to be just right. You need just enough binding, but not too much. All right. Okay, so now we're going to go through this process of clonal selection and diversity. So we've got these, let's say through that process, we've got a B cell. Here it is labeled L, lymphocyte. And it has a B cell receptor. This one is triangle antigen receptor. This is circle antigen receptor here. And maybe this is, if you blow that up, it's actually a star. Only those lymphocytes whose receptor recognizes the antigen will become active. And we've talked ad nauseum a little bit about this, so I'm not going to harp on it. What that's going to do, though, when we think about trying to generate diverse responses, is we need these then to clonally expand, right? So only the lymphocytes that engage with antigen will double. It means those useless ones that are never going to see the light, well, they'll see the light today, but they're never going to actually engage in immune reactivity on your behalf. They're going to stay as either singlets, right, single cells. There's probably more than just one, but they're certainly not going to expand, okay? And this is 2 to the 20. These things can double 20-fold, right? And so 1 turns into 2, turns into 4, turns into 8, turns into 16. You can see how this expands really, really quickly, right? And we'll talk a little bit more about this clonal expansion here in a second. All right, so now we're going to have antigen-dependent differentiation. So once that antigen binds, we just talked about that, to an antigenically committed cell, that cell divides, producing many fully active effector cells. This is that word again that stumped us, some of us. Effectors, just like effector proteins, mediate the response, right, of the initial event. Effector cells in the immune system are going to be the ones that mediate the action of that differentiation event. So the effector cell of a B cell would be what? Plasma cell, right? So this is that cell that becomes differentiated, and remember, it packs itself with a lot of ER, and it's just a professional biomanufacturing facility. Produce antibody X as much as I can and secrete it, right? So the plasma cell is going to be the effector cell. So there it is. The effector cells for B cells are plasma cells. They're basically manufacturing facilities. All right. The effector cells for T cells come in 3 main flavors. The first is the cytotoxic T cells, TCs, kill self cells that are producing foreign antigens, right? So that's that CD8. These are the CD8 positive cells. And their effector cell is technically the cytotoxic T lymphocytes. I know, or CTLs. Right? So before they've differentiated, before they become activated, they are TC cells, cytotoxic T cells. After they are activated, they become CTLs, or cytotoxic T lymphocytes. Okay? Helper T cells induce other cells, right? So this is through production of cytokines, right? The interferons and interleukins, okay? Including their impacts on cytotoxic T cells and B cells to differentiate and proliferate, okay? So these are all, I should say, these all originate as T lymphocytes or T cells, and depending on their expression of CD8 or CD4, they will differentiate down these lineages into cytotoxic T cells or into helper T cells. Okay. The other are these regulatory T cells. We're not going to talk a lot about them, but in this case, regulatory T cells again originate from the T cell population, and in this case, they actually suppress the immune response for common antigens. And this is, so you can imagine you have been born. I'll give my daughter's the example of my daughter. I use her a lot. The first time that she ingests peanut, that is the first time she'll ever experience allergens, right, or antigens associated with that particular product. Right? So everything for a baby is the first, right? So everything is this opportunity, right, to generate an antibody response or a T cell response against these sort of antigenic products. In the case of food, we don't want to generate an immune response against the things that we eat. And so packed in your oral cavity are these regulatory T cells, as well as in your gut, that express both CD4 and this other cluster of differentiation protein, CD25. And these are going to help us develop a tolerance against these types of food-based allergens. All right. So let's talk really quickly then about, just like in talking about immune pruning, we use the T cell as the example. In this case, for differentiation of mature naive into mature effector cells, we're going to take the example of a B cell. Okay, so step 1, antigen bind surface IgG. This is maybe that IgD form, right, of immunoglobulin, or the IgM, which is that initial receptor. in combination with cytokines from helper T cells, right? So that, remember we talked a little bit about that feedback loop, right? So B cells engulf, right? They recognize, they engulf, they digest, they present, they get a T helper cell response that helps stimulate them and say, yes, this is a positive. Please differentiate into an effector cell. The B cells will be activated, okay? So helper T cells will help that. And what they're going to do is they're going to proliferate. We talked about that. Clonal expansion. Divide, 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 right? Please generate a response. And they're going to differentiate. So B cells can make secreted IgGs. They're just really lousy at it, right? They're unequipped to produce the amount of antibody that you want. And so B cells are going to differentiate into two effector cells. The first is the plasma cell. That's the main one. And these are going to make surface and secreted IgG, right? Just make antibody. The other is that they're going to differentiate into what we call memory cells. So the three tenets of the immune system, both innate and acquired, were specificity, diversity, and memory, right? And this is where memory is going to come in, okay? So we need memory cells to help remember or record the fact that this is an antigen that we've been exposed to, or a virus or a bacteria that's producing an antigen that we need to respond to, okay? All right. So we're going to generate, in the case of B cells, but also T cells, primary and secondary immune responses, okay? So this is where those memory cells come into play, right? So a B cell gets activated, make an initial response, right? Differentiate into a plasma cell, produce antibody, but also produce some memory cells so that if I get a second exposure, I'm more ready for the second event, okay? So in addition to the effector cells, memory cells are going to be generated during antigen-dependent differentiation. So we get a first response, right? This T cell now differentiates, proliferates, right? So we get more of this T cell, right? These are, let's say, the effector T cells, right? The downstream effectors. And it's going to generate some memory cells as well, okay, in purple. What that allows us to do is, because these doublings are huge, right? I said 10 to about the 20. We don't want that many T cells or B cells like circulating in our bloodstream all times, right? Like if you can imagine, if you added that many immune cells to immune system over a lifetime of pathogen exposure, right? Your blood would be thick, right? You just would not have enough volume. So what we want is for these T cells, these effector T cells, to go away, but we recorded a memory, and we've turned that one cell into maybe 3 cells, right? It's just an example. These are not real numbers. Okay. So now what happens is the memory cells can provide for an enhanced secondary response. So we get the pathogen again, right? And now we've got a threefold, right? In this particular case, a threefold excess of the original response, right? Because we have more of those memory T cells that are primed and ready to respond to that particular antigen, right? Okay, so memory cells are going to provide sort of persistence of a long-term response, right? Some may last your entire life, right? Some of them may last your entire life until you die. Others may go away, right? So this is the premise of when you get a vaccine, why you get boosters, right? You get those shots when you're 13 years old and you're like, why on earth am I getting another shot? Didn't I get this as an infant? And the reason is that you're trying to reprime that immune response, okay? If you're really trying to generate robust vaccination against pathogens, oftentimes like Hep C, you have to have 3 shots. You get 3 boosters, or 2 boosters plus the original. And so it's only after that 3rd booster, and what they'll do is they'll monitor your antibody response. They'll ask a question which is like, how many antibodies are you making? If you're not making a certain titer, if you're not titering high enough, then you'll get another booster. in order to drive your antibody response up. Any questions? All right. All right, so let's circle back then to Guillain-Barré. Right? So it's an autoimmune response, an autoimmune disease, and we talked about how autoimmunity might originate, right? In perhaps ineffective immune pruning. Chances are you would not live long enough if you have an autoimmune disease at that earliest of age, right? So there have to be other mechanisms to explain why you're happy and healthy, and then at some point in your life you generate an autoimmune response. So we'll talk a little bit about that. None of these are in blue. These are all just other examples of autoimmune diseases and their prevalence. So celiac disease is an autoimmune attack on the intestinal wall. It's exacerbated by ingestion of glutens, right? Type 1 diabetes, we'll talk a little bit more in future lectures about type 2 diabetes. Type 1 diabetes is an autoimmune disease, right? So you've mounted an attack against your beta cells in the pancreas, and if you kill them, then you can't produce insulin and you can't respond to sugar the right way. Multiple sclerosis is, again, a slowly progressive disease of the brain and spinal cord, not attacking Schwann cells, but another component of the nervous system. And rheumatoid arthritis is usually older folks start to develop this, and this is when you develop autoantibodies against your joints. A lot of times it happens to be collagen antibodies that originate, but you can get antibody responses to a lot of extracellular matrix and joint-related proteins. So, all right, so a possible etiology of autoimmune diseases in these cases is a concept called molecular mimicry, right? So if you have two molecules, I introduced this idea in the beginning of this lecture that T-cell and B-cell receptors are going to bind one epitope or other epitopes that are extremely similar, right, in their structure. And this is one of the concepts of molecular mimicry, where you have an antigen that is going to initiate some type of immune response, but that antigen receptor, B-cell receptor, T-cell receptor, also recognizes something that's very, very similar. All right, so we're going to result in an antigen receptors that, quote-unquote, cross-react. This is not a chemical reaction, it's just a binding event. But cross-react means that the protein, in this case the antibody, let's say, can bind two things that are different but similar enough. And antibodies are notorious for cross-reactivity, right? And so certainly those that we produce more on a biotechnological scale. All right. Okay, so if we generate antigen receptors that combine with self-proteins that look a lot like or mimic the antigen, then we get an immune system response. And now, if we continue to mature, now you've got that autoantigen rifling through your body, right? And it looks to the immune system that your body is an infection, right? And so you're getting copious amounts of that motif being priming the immune system. And now the self-protein may be inhibited if that protein has a specific function, like a growth factor. Maybe you've inhibited the ability of that protein to bind and engage its receptor. Or you can have the cells that are expressing that antigen negatively affected, right? If I've generated a cytotoxic T-cell response, then my T-cells will start attacking the cells, right, and killing those cells that are expressing that protein. And remember, those autoantigens, right, every single one of your cells has an MHC class 1 receptor system, right? And so you're always presenting your self-peptides as well to the immune response. All right. So in Guillain-Barré, most cases begin about 1 to 2 weeks after an uninfection, right? And that infection is this C. jejuni. It causes enteritis. So we talked about colitis, infection or inflammation of the large intestine. Enteritis is inflammation of your small intestines. Okay? So you get some sort of inflammatory response, and this is thought to be an instance where mimicry is the reason. So what is the antigen? How's it presented? And is the immune response humoral or cell-mediated? And what is the autoantigen? We're going to talk about that. All right. So we've got about 10 minutes, and we should be fine to get through this. So I'm going to throw you guys a curveball now. I apologize. So I want to talk real quickly about this protein called CD1. This is another one of those cluster of differentiation proteins. And this was the first one ever discovered, which is why it gets the, I guess, the honor of being called CD1. And what this is it's a antigen-presenting protein. It's not an MHC, but it's an MHC-like protein. Okay? Instead of presenting peptides, what CD1 does is it presents lipids. So just like we can bind peptides and display them. In this case, we're going to bind up lipids that are associated with what in this case? In this case, it's a bacterial lipid, right, from that bacterium called C. jejuni. So the pathogenesis is we get infected by C. jejuni. It gets taken up by the B cell. The B cell does what it does, right? So the B cell has recognized some antigen on C. jejuni, right? And that is what's inspired it to bring it into the cell, right, to phagocytose it. It's going to chew it up, and now it's going to present on, it's a professional phagocyte, right? So a professional antigen-presenting cell. So it's on MHC class 1s. If those proteins get out into the cytoplasm, it'll present those. Through MHC2 class proteins, it will present the bacterial proteins that it's digested. And it will also present lipid moieties using this CD1 protein. Okay? So that B cell will present all those things to as many T cells as it possibly can. And when it finds, if it finds THC2 presenting, if it's presenting this protein or this, good grief. Let me just hit, do I get the easy button? I'm going to hit restart. Lipooligosaccharide, so this is a lipid sugar moiety on CD1. You have a subclass, I mentioned this earlier to a question. You have subclasses of these T cells. In this case, you have what's known as a CD1 restricted TH cell. So these are only going to express CD1, the cognate receptor that'll recognize CD1. Okay, and as a consequence, it's going to do what TH cells do. It's going to make all those cytokines, right? It's going to positively feed back into that B cell, right? And if the B cell receptor that recognizes C. jejuni is the receptor for a lipooligosaccharide, then what you're going to do is wind up producing antibodies against lipooligosaccharide. All right, so that's the epitope that's recognized by the B cell. It is presented then to the T helper cell, and it generates an antibody response. Okay, so it's a humoral response. The autoantigens are these proteins called gangliosides. And so the issue is that we express lots of gangliosides. This is in this broad class of lipids called sphingolipids, and these are really important molecules in proteins and cells that have a heavy amount of signaling. Right? So gangliosides are enriched in membranes where signaling occurs. They primarily cluster these things called lipid rafts, which we've, I think, introduced at least at a high level. And the problem is that parts of their structures, in this case, these sugar moieties on ganglioside GM1, is almost perfectly identical to the GM1-like C. Jejuni protein, or excuse me, lipid that's presented. All right. So now I'm going to produce antibodies. I'm meaning to produce an antibody against this species, but that antibody has cross-reactivity now to my own molecules. I think I keep saying protein. I apologize for that. All right. So we're going to generate an autoimmune attack on those gangliosides, and for reasons that are still really unknown, the cells that seem to be affected more than any other are these Schwann cells. Right? And Schwann cells are the cells that are lining these axonal bodies of your nerves, and they're going to produce this molecule called myelin, these myelin sheaths, and they're like signaling insulators, right? So the way neurons, those of you guys that are in physiology, hopefully are talking about signal propagation in nerves. It's an action potential. It's driven down the axon body, and if you don't have this insulator around the axon, that signal diffuses out. It won't go along. It won't continue to travel down the axonal body. Okay. So when we get demyelination, we slow or even completely halt nerve conduction, and as a consequence, all the peripheral nervous, everything that's impacted by peripheral nerves, starts to become impacted. Breathing, our heart rates, flaccid paralysis, right? All these things that we talked about. Okay. Treatments. Well, if you get this, your biggest problem is it's hard to breathe, right? You do not get the sympathetic nervous activation of your diaphragm, right? And so you literally forget to breathe. So in the olden days, they would stick you in what's called an iron lung, right? So this is this really, if you think about the technology at the time, a pretty clever device where this is essentially a pressure chamber, and they pull a negative pressure on the entire body, and as a consequence, it forces you to take air in, right? So this is called the iron lung. This is actually a polio treatment facility. So polio suffers from, patients with polio suffer from similar issues, and these are a bunch of iron lungs. Nowadays, we actually do forced ventilation. So instead of pulling a negative pressure, we push air down. This actually keeps this person alive, but it's actually quite bad to force air into somebody's lung, that we've learned. But I guess you take your poison. Immunosuppression therapy. Obviously, if I can inhibit my immune system, then I can try to quelch the autoimmune response. We can also do something called plasmapheresis to patients. Anyone know what plasmapheresis is? What does it sound like? What's that? Somebody say something? We're going to get rid of somebody's plasma. So the antibody, because it's a humoral response, right? It's mediated by all those antibodies that plasma cells are making. And so if I can take your blood and I can extract the plasma from the red blood cells and replace your plasma with something else, like saline or serum albumin, right? Solutions of serum albumin, which is your main constituent of your plasma. Then what I'm doing is I'm sucking those, I'm effectively removing those antibodies from your plasma and replacing it with fresh plasma or plasma substitute. Yeah?
So you don't replace it with the plasma?
Well, so the plasma donor, if they're a good match, then yes. You have to do this so often that it winds up becoming sort of a supply issue. And so it's a lot easier. There are a lot of groups that are funding blood volume mimetics and blood development of plasma-like products and things like that. And they're used for this. And so if I can get rid of those antibodies, then presumably I can get rid of the response. I can also give immunoglobulin injections to try to inhibit those autoantibodies as well. Okay? So mostly, can we dilute out the response, right? Can we either stop plasma cells from making it, the antibody, remove the antibody or try to find ways to inhibit that antibody. Okay? All right. If you think this is really complicated and cool, we have a lot of really great folks here at UVA, Tim Bender, Vic Engelhardt, and Lauren Erickson that are in the Burns Center for Immunology. And so I encourage you guys to reach out to them.